Alright, welcome back everyone to TMM3104. My name is Dr. Kevin He, and last week we talked about Planaria and how they're able to undergo full body regeneration. As you can see, they're able to regenerate their entire bodies from small portions once cut. This makes them really interesting for regenerative medicine because they have really similar anatomical features and genes as humans. So we're going to show everyone's videos. Just a reminder that 40% of your mark will be on creativity. So good luck everyone. You want to go first?
Yeah, sure.
So today I'll be covering some of the basic mechanisms and cells involved in Planarian regeneration so we can get a better understanding of its application in human research. The Planarian body is stocked with neoblasts, cells that don't have an innate function but rather differentiate based on the body's needs. They are the pluripotent adult stem cells that underlie Planarian regeneration. Neoblasts account for the entire population of adult dividing cells as they are abundant in mesenchyme tissue and divide continuously. This microscopy image uses tissue-specific markers to demonstrate the differentiation of Planarian stem cells into mesenchymal tissue. Upon tissue damage, these widely distributed neoblasts are recruited to the wound site. Outgrowths at the wound site, called blastemas, begin forming the new tissue. A blastema is a mass of undifferentiated cells that proliferate rapidly to replace damaged tissue in a process known as epimorphosis. In cases of substantial tissue loss, a process called morphollaxis will commence, whereby cells in the decapitated stumps are reorganized, reforming a full-fledged organism. This whole-body response is likely regulated by the extracellular signal-related kinase pathway, also known as the ERK pathway, whereby messages propagate throughout the entire body to notify if there has been an injury.
Yeah, that was awesome. Serena, you're up next.
Okay, awesome. I'll just pull it up.
How is whole body regeneration in Planarians coordinated? Regeneration requires two factors in order to function: potential and instructions. We already know pluripotent stem cells provide the potential to produce new tissues, but we can now discuss how PCGs, positional control genes, provide the instructions for controlling regeneration and tissue turnover. PCGs are regionally expressed in the subepidermal layer of muscle cells along various body axes. They provide positional information through the patterning abnormal RNAi phenotype or through the regulation of pathways. Unfortunately, phenotypes have not yet been revealed, so their role awaits elucidation. In regards to pathways, one such pathway is the WNT signaling pathway, which decides whether to regenerate a head or tail at transverse amputation planes.
Wow, that was awesome. Natasha, do you want to go next?
Yep, sounds good.
The question that I've chosen to explore is how Planaria regeneration processes might have implications for human health. Autophagy is a cellular process that involves the degradation and recycling of cellular components crucial for maintaining homeostasis during regeneration in these remarkable creatures. What's intriguing is that autophagy is not only important in Planarian regeneration, but also in human neuronal development. These processes can alter the cytoplasmic composition of cells and influence their fate. The big question is here whether autophagy in humans directly or indirectly regulate neoblasts, the remarkable cells responsible for Planarian regeneration. This is an area where further research is essential. One reason to explore this connection further is the striking structural and genetic similarities between humans and Planarians. Both species share a central nervous system structure, neurotransmitters, and neuronal cells. In fact, approximately 95% of the central nervous system-related genes in Planarians have human homologs. This suggests that a potentially close link between the regenerative mechanisms and our own.
Wow, that was great. Ren, do you want to go next?
Sure.
A very interesting characteristic that Planaria have been explored for is their trained immunity, which is the existence of an innate immune memory for previously encountered pathogens. This involves epigenetic reprogramming through histone modifications that modulate the expression of antimicrobial genes during infection. Some mechanisms that they employ to increase the expression of specific antimicrobial genes target pathogens that also infect humans, including E. coli and S. aureus, and therefore studying these mechanisms may reveal new ways to combat human infection.
Yeah, so as we learn more about Planaria, we're going to learn more about stem cell biology and regenerative medicine, and even cancer research as we learn more about cell differentiation and proliferation. So I think everyone did an amazing job, and everyone deserves 100%, guys.